something in common with these creatures and millions of others roaming there? You are all classified as animals. What does classification mean and how it is used to organize and identify the animals in the world? How many species of animals are there? Scientists sort the different types of animals in the world into categories based on certain characteristics. Animal classification is a system of organization. Animal kingdom classification is an important system for understanding how all organisms are related. Classification means organizing things according to the common characteristics. For example, your school library, you can see academic books on one side, academic magazines on another rack, and reference books for each subject on another section. Another example is your kitchen. Grams, pulses, curry powders, and vegetable oils are categorized and stored in separate containers. All these categorization make the identification process easier. So, to classify animals, scientists developed a system to categorize and identify animals according to their common traits. Let's take a quick review of the different bases of animal classification. Classification of animal kingdom is based on various fundamental features like levels of organization, symmetry, diploblastic and triploblastic organization, coelom development, segmentation of the body, and presence or absence of notochord. All members of Animalia are multicellular heterotrophic eukaryotes, but all of them do not exhibit the same level of organization. The cells in their body are of several types. These are organized into several functional units of progressively increasing complexity. The animal body shows 4 basic levels of structural organization as given below. The first one is the cellular level of organization. In sponges, many cells arranged as loose cells aggregates, and the body shows some division of labor among cells. They are remarkably independent and can change their form and function, but the cell do not form tissues. This level of organization is known as cellular level of organization. Coelenterates shows another kind of organization called tissue level of organization. Here, the coelenterates are like jellyfishes. The arrangement of the cell is more complex. The cells performing the same function are arranged into tissues. Hence, it is called the tissue level of organization. The next type of organization is the organ level. In Platyhelminthes and other higher phyla, tissues are grouped to form organs, each specialized for a particular function. That is, organ level of organization is present. In animals like annelids, arthropods, mollusks, echinoderms and chordates, organs have associated to form a functional system. Each system concerned with a specific physiological function. This is called organ system level of organization. Organ systems in different groups of animals exhibit various patterns of complexities. In humans, excretory, respiratory, circulatory and reproductive systems are present. And the digestive system and circulatory system of octopus are different from other organisms. For example, the Platyhelminthes shows the organ system level of organization, but the digestive system is incomplete and the circulatory system can also be open and closed type. Let's try to classify animals according to their body symmetry. Symmetry refers to the arrangement of parts on the opposite side of the body of a three-dimensional animal. Based on the symmetry, animals can be asymmetrical and symmetrical. The symmetry may be bilateral or radial one. Let's discuss the symmetry of organism in detail. First, we will look into asymmetry. Here, you can see that in sponges, any plane passing through the center does not divide them equal halves. Such animals are called asymmetrical. The body of some animals can be divided into two equal halves by one or more planes. Such animals are called symmetrical. The symmetry can be further divided into two. The first one is radial symmetry. When any plane passing through the central axis of the body divides the organism into two identical halves, it is called radial symmetry. Coelentrates, ctenophores and echinoderms shows this type of symmetry. The next type of symmetry is bilateral. Animals like annelids and arthropods, the body can be divided into identical left and right halves in only one plane. This is called bilateral symmetry. Animals are classified on the basis of germ layers also. Let's explore how. Germ layers are groups of cells behaving as a unit during the early stages of embryonic development. It differentiates to give rise to all the tissues or organs of the fully formed individuals. Based on the germ layers present during the embryonic development, animals are classified as diploblastic and triploblastic organisms. As the name suggests, the cells are arranged into two embryonic layers in diploblastic animals. As you can see in the image, external ectoderm and the internal endoderm present in diploblastic animals. In addition, an undifferentiated layer, mesoglia, is present in between ectoderm and endoderm. Coelentrate shows this type of organization. Then, what do you mean by triploblastic? Yes, the developing embryo has three germ layers: the outer ectoderm and inner endoderm, and a third germinal layer, mesoderm, present in between the ectoderm and the endoderm. All animals from phylum Platyhelminthes to phylum Chordates are triploblastic. Likewise, a principal body cavity in most animals located between the intestinal canal and the body wall is known as a coelom. Let us see how animals are sorted on this feature. The body cavity present between the body wall and the gut wall lined by mesoderm is called coelom. The presence or absence of coelom is very important in classification. On the basis of coelom presence, animals can be classified into three different groups. The first one is acoelomates. Here, you can see that the body cavity is absent. Such organisms are called acoelomates. Platyhelminthes is an example of acoelomates. The second one is pseudocoelomates. Pseudo means false. So, the pseudocoelomates contain a false coelom, which means in some animals, the body cavity is not lined by mesoderm. Instead, the mesoderm is present as scattered pouches in between ectoderm and endoderm. Such a body cavity is called pseudocoelom, and the animals possessing them are called pseudocoelomates. Aschelminthes are pseudocoelomates. The animals having true coeloms are called coelomates. A true coelom arises within mesoderm and is therefore lined by mesodermal tissues, that is, externally by parietal peritoneum and internally by visceral peritoneum. Furthermore, in some animals, the body is externally and internally divided into segments or somites, with a serial repetition of at least some organs. Segmentation can be of following two types. A segmentation that simultaneously divides the body both externally and internally is called metamerism or metameric segmentation. This kind of segmentation is found in annelids, arthropods and chordates. The other kind of segmentation present in animals are false segmentation or pseudo-metamerism. It is found in tapeworm. The body is divisible into parts or segments externally, and each segment is independent of the others. These segments are called proglottids. Not only body segmentation, presence or absence of notochord also differs animals. What is a notochord? During embryonic development, a mesodermally derived rod-like structure is formed on the dorsal side of the body in some animals, and it is called the notochord. Based on the presence and absence of notochord, animals are divided into chordates and non-chordates respectively. Scientists have developed a classification system that categorizes all members of the animal kingdom. We explored how the classification of animals is done based on their different features like body plans and anatomical complexity. Let's explore the broad classification of kingdom Animalia based on common fundamental features in the upcoming session. Now, let's summarize what we have learned.
Manorama Horizon. Learning made simple.